Welcome to Cancer Lesson 5.2, What do cancer screens really tell us? The goals of this lesson are to explore the concepts of sensitivity and specificity in preventative screens and consider the challenges of using population statistics in medicine. At the end of the lesson, the students should be able to define the terms sensitivity and specificity and false positive and false negative with respect to cancer detection and diagnosis. Describe the limitations of mammograms and the PSA test with respect to cancer diagnosis, and describe the challenges of making individual decisions based on population-wide evidence. We will achieve these goals by engaging in a Socratic discussion and a group activity in which students evaluate hypothetical cancer screens. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. The purpose of preventative screens is to identify people with the disease but who do not yet show any symptoms. Screens are not 100% accurate due to false positives, which is when a patient tests positive but does not have the disease, and false negatives, which is when a patient tests negative but does have the disease. The sensitivity of a screen is how well the screen detects people who have a disease, and the specificity of a screen is how well the screen excludes people who do not have a disease. You can review the scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure, and the student workbook provides additional explanation for students. Be sure to print the lesson worksheet and the homework worksheet, which is an information handout located in the Lesson 5.3 curriculum materials. The key points of the do now are that screens identify abnormalities in healthy people but are rarely diagnostic on their own. How good a screen is depends on its sensitivity and specificity. The sensitivity of a screen is how well the screen detects people who have the disease, and the specificity of a screen distinguishes between people who have and don't have the disease. The do now begins by reviewing yesterday's homework article, in which the author contemplates the dilemma of using evidence that is based on population statistics to make decisions about her personal health. Next, students discuss the characteristics of an ideal screen. This leads to a discussion of what would make a screen less than ideal, which leads to defining the key terms of sensitivity and specificity. The key points of the discussion are that screens are used to identify abnormalities, and screens are not used to confirm what the abnormality is. The sensitivity and specificity of screens are trade-offs. When one is improved, the other is decreased. False positive results can lead to overdiagnosis. In this population of 10 people, 6 are healthy and 4 have some kind of abnormality. An ideal screen would detect all the healthy people as healthy, the green smiley faces, and all the people with abnormalities as unhealthy, the orange smiley faces. Note that these slides are animated to help with the explanation. A screen with sensitivity problems would not identify all the people in the population with abnormalities. Instead, some of them would be told they are healthy when they are not. In this example, 40% of the population have the abnormality, but the screen has only identified 20%. Put another way, the screen is missing 50% of the unhealthy people. It is said to have a 50% false negative rate. As a result, 20% of the people in the total population won't get the additional diagnosis and treatment they need. Conversely, a screen with specificity problems will tell some people who are actually healthy that they have the abnormality. In this example, 30% of the population are told they are unhealthy when they are not. Put another way, 42% of the positive results in the screen are false, a 42% false positive rate. So 30% of the people in the total population may be treated for a disease they don't have. A false negative result occurs when a screen doesn't detect disease in someone who has it. A false positive result occurs when a screen tells someone who is actually healthy that they are not. It is obviously a problem if people who are sick don't get follow-up diagnosis and treatment, but false positives can also result in the problem of overdiagnosis. Next, we run through 2 real-world examples of the impact of false positives: imaging tests that screen for breast cancer by detecting dense tissue that may turn out to be either malignant or with the potential to become malignant, or the blood screen for prostate-specific antigen that is used to screen for the presence or potential of prostate cancer. In both cases, a positive screen result, either dense breast tissue or elevated PSA levels, gives no information about whether the abnormality will progress to a life-threatening disease. The confirmatory biopsies that follow up these screens are very invasive and in some cases lead to severe side effects, like incontinence in the case of a prostate biopsy. The third type of screen in common use is a genetic test. The example given here is the BRCA test that is used to detect inherited mutations in the BRCA proto-oncogenes that increase the likelihood of developing severe forms of breast and ovarian cancer. It is important to note the distinction between inherited and non-inherited mutations. Mutations are only inherited if they are found in egg cells and sperm cells. They are called germline mutations. Most genetic screens look for inherited germline mutations. In contrast, mutations to the tumor cells themselves cannot be inherited. They are called somatic mutations. In the activity, students will work in small groups and evaluate the utility of 2 hypothetical new screens for liver cancers and carcinomas. Students should answer the questions on Worksheet 5.2 that will prompt them to consider which screens are appropriate for certain patients. They will also compare their attitudes about the screens to government policy. Note that the false positive and false negative percentages listed in the activity are invented. The key points of the lesson's wrap-up are that the results of screens are described as population statistics. While population statistics can define trends well, they do not mean that the individual person will behave like the average person. In the wrap-up, students will use their worksheets to focus on the consequences of false positives and discuss why the evidence from population statistics may not always be valid for an individual patient whose behavior is not average. The tension between the data provided by population statistics and an individual's behavior is at the center of many cancer treatment dilemmas. Often, the treatment available to individuals is limited by resources. For homework, assign each student one of 3 specialties: surgeon, radiation oncologist, and medical oncologist, and give them the appropriate 2-page informational sheet that they should read for tomorrow's lesson. One of the most common questions students ask is, how would I know whether a DNA sequence has mutated? It's important to have a reference sequence to compare it with. In the case of germline or inherited mutations, every cell in the body will have the same mutation, so the reference sequence must come from someone else. In the case of somatic mutations, only the tumor cells are mutated, so the reference sequence can come from another cell in the body, usually a cheek cell. At the end of the lesson, collect the lesson worksheets to assess students' ability to evaluate the utility of different screens. This lesson connects to Lesson 5.1, which looks at screening at the individual level by giving the population perspective. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.